for inviting me. And I brought some flowers with me, even so there are many here. I realized, but Rajiv, these are very expensive flowers from Van Gogh. And I want to use this slide to congratulate the Center of Excellence in Genomics here at ICRISAT for the excellent work you have done, outstanding, groundbreaking research, illustrated by many awards that you obtained, and of course the outstanding number of publications. I will talk about a new tool that can be used, I think, in many crops, namely use of double tablet libraries developed from Landraces. And my talk will cover three aspects, namely the production and molecular characterization of double tablet line libraries. Second, I will talk about the agronomic evaluation of this material in comparison with elite material. And finally, I will mention a few applications where these libraries can be used in genetic research. Okay, we have seen this slide already in various other settings. Why are we interested in Landraces? And here you see, I've used the example here of European maize, but my remarks would apply to many other oleaginous crops. Until the 1950s, thousands of Landraces have been cultivated by farmers in Europe only. And with hybrid breeding, only about, I would say, the foundation of the Flint heterotic pool traces back to between 10 and 20 founder lines that have been developed by not more than 5 Landraces. And that means, well, and this material was, of course, then used to establish the elite heterotic pool. And obviously, with this approach, we have captured only a very small part of the rich genetic variation present in the Landraces. So the conclusion is the elite germplasm has a very genetic base. There is pervasive evidence from molecular studies about this. And this holds true especially, I think, for many agronomic traits that may become very important in the future to cope with the challenges, for example, of climate change and to develop new bio-based products. Okay, there are thousands really of Landraces stored in seed banks, and I give here the example of maize, for example, at CIMMYT. They store in their seed banks 25,000 accessions just from Landraces, and this holds true for other seed banks as well. So the conclusion is there is a huge number of Landraces stored in seed banks, and I would think, maybe that's a bold claim, 99 more, I would say even more than 99% have never been used in breeding. Okay, now how can we use this material? We have to be aware when we want to mine the Landraces for hybrid breeding, for example, there is a huge gap, a huge canyon between the Landraces and modern germplasm. And with continuous progress in breeding, this canyon gets even bigger. Now, Landraces are heterogeneous populations of heterozygous individuals which have a relatively large genetic load. On the other hand, hybrid breeding rests on development of homozygous lines that have acceptable per se performance and that have hopefully very good general combining ability. So now the question is, how can we bridge this gap? And there is an old bridge available, namely recurrent selfing. And I will show that this was not a very effective bridge. And nowadays, with a double tablet technology, we have, I think, a new tool available, a new bridge that is very promising. Now, before I talk about this bridge, let me briefly explain to those of you who are not familiar with the DH technology in maize. For example, if we have here a plant from a landrace, we would pollinate this with pollen from this inducer line, which is usually terrible in agronomic performance, but it has a nice property that the seeds that you harvest after pollination, about 90% of these seeds are normal crossing seeds, whereas about 10% are haploid seeds. And if you have a suitable marker system, for example, here an embryo marker or an oil content, you can distinguish the haploid seeds from the crossing seeds. And the haploid seeds are then germinated. They're treated with colchicine to double the chromosomes, and then if everything goes all right, you obtain a doubled haploid plant, which you then self-pollinate, and then you have in one step already fixed inbred lines. Okay, now if you apply this procedure, and let's now first start with the conventional procedure, namely recurrent selfing. You see here the number of surviving lines under inbreeding. At the beginning, we have a very large number of lines, and as you continue with selfing, the number is really gradually decreasing, and you end up with a very small number. Due to the high inbreeding depression, you lose many of the lines because the genetic load is uncovered gradually, stepwise. And of course, during this phase, you spend a lot of your resources. Now, when you develop double haploid lines, you go in one step from this A0 plant to a fully homozygous plant. Of course, you have also to cope with the entire inbreeding depression, but the advantage is that it's done in one step. So if you start with a large number of haploid seeds at the beginning, there is hope that you end up with a sufficient number of reasonably good double haploid lines. So this was our idea. Now, what are the expected advantages when you develop double haploid line libraries? We obtain immortalized genotypes in one step because we have now fixed lines available, and these lines, they can be identically multiplied by selfing, and once we have inbred lines, they can be phenotyped with very high precision in replicated trials. That's, of course, very different from if you have only individual plants available from a landrace. So there is much better characterization of the accessions, and I think this is very important for breeders because then they have really access to well-characterized genetic variation. Now, the genetic variation is fixed, so it's much easier to conserve this material. It can be regenerated easy, and it's controllable because I think detecting an outcross, for example, inbred, is relatively easy. That's certainly an advantage for seed banks. So regeneration is easier and controllable. And another advantage is that with double haploids, you have the total genetic variance directly available. Namely, you have the variance among double haploid lines is twice the additive variance, whereas if you would look at the original landrace, the variance among A0 plants is only the additive variance, so that's twice as much variance. And another advantage could be that the landrace is purged, I would say partly purged, from the genetic load. So now let's also look at some critical questions. How should we identify the most promising landraces? I've mentioned we have thousands of them stored in seed banks. So how, and of course this procedure is there's no free lunch, so it's relatively expensive, that we'll see. What are the success rates for producing such libraries? How expensive are they? And what is the effects of purging from the genetic load, not only at these loci that are directly responsible for the genetic load, but for those that are of agronomic interest? And finally, the question is, are double-thread line libraries a faithful representation of the landraces in terms of the allelic inventory and the linkage disequilibrium? Okay, in the first part of my talk now, I will talk about the production and molecular characterization of double-tablet line libraries. And here you see the roadmap, how we approached or looked at the problem. We evaluated the test cross performance of 70 landraces. We had also to check for expression of the embryo marker system that we employ for distinguishing the haploid seeds, identifying the haploid seeds. And after this step, we produced double-tablet lines from the 10 most promising landraces identified in this step. In other words, we produced double-tablet line libraries. We evaluated the per se performance of these double-tablet lines from 6 of these landraces, and currently we are evaluating the test cross performance of 79 selected DH lines identified in this step. So this is still going on. Okay, here you see now the results when we evaluated the test cross performance of 70 landraces, and we compared them with the performance of 5 hybrid checks. Now here you see the distribution for grain yield, our number one trait, obviously. This is a distribution of the test cross performance of the landraces. It ranged from 7 tons to 10.5 tons per hectare. Compared to the elite checks, there was a yield gap of 26% on average, but the best landraces are only 17% distant from the elite checks. Yeah, the next question was, what is now the success rate when we produce double-tablet line libraries from landraces? And we compared the success rates now to the success rates we have in elite material. So we compared 5 landraces in their success rate with 9 elite crosses that we would usually use in our breeding program for developing double-tablet lines. We divided the entire process in various phases. First, I should mention, we produced, we had 1,000 plants from each landrace in the induction crosses. In other words, we produced 200,000 seeds, and about 3% of these seeds were identified as haploid seeds. So these seeds were then germinated, and then we recorded the number of haploid or double-tablet plants in the field. And when we took now the ratio of this, the number of haploid double-tablet plants obtained from the number of germinated seeds, this gives the survival rate in during this phase, where we have colchicine treatment and so on. Okay, and the next phase is the development of seeds in the D1 generation. The number of lines that show seed set obtained from the number of haploid or double-tablet plants, because you do not always get seed set. Some of these lines or plants are sterile. Oftentimes they are male sterile. So this is what we call the reproduction rate. And now let's look at the comparison. For the survival rate, this phase, there was not a big difference between the landraces and the elite material, at least to my surprise. Now, when we look at the reproduction rate, actually the reproduction rate was twice as high, more than twice as high in the elite material compared to the landraces. So this is the problem, in other words, the poor reproduction rate. And when you now multiply these two rates, you obtain the overall success rate of the procedure. And the overall success rate in the landraces was about 1/3 of the success rate in the elite material. And this leads to the conclusion that the cost of producing a double haploid line from a landrace is about 3 times as expensive as the production of a line, a double tablet line from elite materials. Okay, and the next question we had, do we do the allele frequencies differ between our original landrace and the double tablet line library that we obtained from it. And to make this comparison, we genotyped the landraces, we genotyped always 23 S0 plants from the landraces and compared the profile with the profile of the double tablet lines obtained from the respective landrace. And here you see now the results of the FST statistics, where we compared the allele frequency in the original landrace with the allele frequencies in the double tablet line library. And only in a few cases we found indeed significant differences. That means there are not big changes in the allele frequencies. Also, you see the same picture now when we looked at the cluster analysis. And here you see the trees. They look almost identical between the original landraces and the double tablet line libraries. And the same holds true when we look at the decay of linkage disequilibrium in the original landrace and the double tablet lines derived from them. So to summarize this first part, we could show a proof of concept for the production of double tablet line libraries, but the price is about 3 times as high compared to elite materials. And we could demonstrate that the libraries captured the molecular diversity of the landraces faithfully. The difference in allele frequencies were small, and the LD decay was similar. So the conclusion is the double tablet line libraries, they represent immortalized gametes of the landraces, and they are an alternative for conserving landraces in seed banks. I cannot cover this aspect. I think we can conclude that genetic load that is present in the landraces is mainly due to is of polygenic nature. Now let's come to the second part, namely the agronomic evaluation of these libraries in comparison with elite material. And here you see now, this was done with only 6 landraces. And here you see the number of lines that we have obtained in the libraries. They vary, even so we started always, to remember you, with 1,000 of plants that had been used in the induction crosses. In other words, there is quite a substantial variation in the success rate between landraces, here from 21 DH lines up to 102 DH lines. And these lines were now evaluated in multi-test trials and compared with the performance of elite flint lines from our breeding program. Yeah, and we conducted now an analysis of variance to look at the variance among the double tablet line libraries and within the double tablet line libraries. Here you see the various traits that we looked at, starting with emergence, early vigor, up to grain yield. And what we can see on average, there is only 20% of the genetic variation is among landraces, 80% is within landraces. That has very important consequences. And here you see now the results of an analysis of molecular variance showing essentially the same result, namely 20% of the molecular variance is among landraces and 80% is within landraces. When we look at the performance of the libraries here for grain yield, the libraries do not vary very much in their mean. They vary a lot, however, in the genetic variance within the landrace. And for that reason, we looked at the usefulness criterion. The usefulness criterion is calculated as you sum up the mean, which you see here, and you add the expected genetic gain, and here we use the selection intensity of 20%. In other words, what would you obtain if you would select 20% of the best DH lines? And for example, in this landrace Satomare, the usefulness criterion was very good. It was, of course, not as high as with the elite material. The average performance of the Landraces was only 50% compared to the elite material. But now let's look at this outstanding Landrace. You see the mean is, of course, much lower compared to the elite material. But when you look now at the genetic variance, it's much higher. And some of the best lines from this Landrace have about the same yield as the elite material. Yeah, so to summarize this part, we find generally a much higher genetic variance within the Landraces than between. And the conclusion is now it's much better to sample a few Landraces with a large number compared to producing double tablet line libraries from many Landraces. There is obviously a gap in the performance between these libraries and elite materials, so there are special pre-breeding programs required. And the recommendation is to produce the double tablet line libraries in excess so that one can eliminate more than 50% of the poorest DH lines before multiplication. This is, of course, a recommendation for pre-breeding, not for seed banks. Okay, now let's look at the third part, use of double tablet line libraries for genetic research. And I have mentioned the strong LD decay in the Landraces, for example, here. That's much more rapid compared to elite materials. And that means this material should have really excellent opportunities for genome-wide association studies. It should provide you with a very high map resolution. And we have started to do this. We looked at the material that I just described in a genome-wide association study, looked at various traits, and for example, for early vigor, we found a very clear signal here in chromosome 5. For fusarium resistance, we found signals at various other locations. For seed set, we found quite a large number of signals again, and actually I think this is very interesting because this is most likely related to the genetic load. And for oil content, we found also a very clear signal. And we are presently looking now at candidate genes, and we found already very interesting aspects. And I should also you should pay attention to our QQ plots. They're really almost perfect. There is practically no inflation. So this shows, again, this material should be excellent for genome-wide association studies. So to summarize, the libraries from Landraces offer a new tool for conserving genetic resources. They are a much better means for characterizing the breeding potential of Landraces, and they are a new tool for genetic research. However, big investments are needed to lift this tremendous treasure for future plant breeding. And before I close, now I've talked a lot about maize. What about other partially allogamous crops? Can this method be applied to other crops? Now, first I have to mention that the DH method is well established in various other crops such as sugar beet, rapeseed, and numerous vegetable species. And there is hope that the system can be applied in other crops soon because there are publications available, for example, in Nature, on production of haploids in Arabidopsis. And this year there was a publication about cloning of the gene underlying haploid production in maize. And of course, with the new technologies, with CRISPR-Cas available, I think there is big hope that we have soon this technique available in other crops as well. Okay, now my proposal, I've mentioned the big investments, and for that reason, to cope with this problem, I want to propose a public-private partnership. Genotyping and production of double tablet line libraries could be done by seed banks, I think, and institutes, as well as CGIAR centers such as ICRISAT here. Now, the phenotyping often, I think, is the bottleneck because it has to be done in multi-location trials. And there I would invite breeding companies, institutes, and of course CGR centers as well. Okay, I have to acknowledge the contributions of quite a number of persons. First, Wolfgang Schiprak, our breeder who developed the material, and the characterization was done together with our team of technicians. Franz Mauch is one of the leading persons, and Juliane Böhm was involved in the computation, and we got financial support from these two projects funded by the Federal Ministry of Education and Research, and both projects have been initiated and led by Chris Schön. And I think this is all. Thank you very much for your attention. Yeah.
Thank you, Dr. Melchinger, for providing us the usefulness of double haploids to enhance the use of the germplasm lines as such. So we'll have a few questions. Yeah, I have 2 hands, 3 now. Yeah, we will do this and then try to.
Mine is not a question. It is basically a expression of my interest. I am from Indian Council of Agriculture Research, and we have initiated some work on double haploid system in maize. And probably what is my request, how we look for partnership with you, if you we can take it forward. If
could you repeat the last part?
I am from Indian Council of Agriculture Research.
Yeah.
And we have initiated some program on double haploid production with the inducer system. And
Yeah.
it will be great if we can have partnership to take some project together, and then we can initiate.
Yeah.
I mean, I was very happy seeing your presentation.
Yeah.
And that is the only thing I just wanted to
Yeah.
say.
Thank you very much.
Thank you.
One of potential partners is sitting here, namely Dr. Prasanna. We had with CIMMYT, I think, quite a long-standing relationship on the DH technology. Actually, we had a very nice program funded by the Gates Foundation where we developed tropically adapted inducer lines that are available now, second generation. And we have continued in this respect. We looked at various alternatives, for example, to colchicine treatment. We are looking for alternative marker systems, so this is still going on. But I think, Dr. Prasanna, we can say this was very successful research. And again, I think there is a possibility for you.
Thank you. Yeah, we have one there. Second one, Dr. Sujay Rakshit, and then.
Okay. This is Ramu from Cornell. So you mentioned that double haploid is helpful for precision phenotyping. So did you ever try this method in teosinte to better understand the domestication events in maize?
Yeah, of course. We were thinking about looking also at teosinte, but for me that would be the next step. You know, we were happy to see that the method works in Landraces. And as I've said, 99% of the genetic variation that is stored in seed banks, collected with big efforts, maintained with a lot of money, is really not used there. So for me, the most obvious choice to broaden the genetic diversity is looking at Landraces. The next step, and there may be certain traits where we do not find the genes in the Landraces, would be to look also at teosinte. Now in teosinte, I think the selection of haploid seeds might be a little bit more difficult. And most likely, Prasanna, we should look at that. I think our oil system might be something which could help.
Thank you. Maybe you said this and I didn't catch it, but so how often were you able to detect deleterious alleles that were basically lethal as non-propagated if you took a particular plant from the land race and recurrently crossed and selected a bunch of doubled haploids that derived from the same parent, and then when you mapped the resulting plants, you found that particular alleles were depleted. I mean, did you actually see that there were particular loci that were never, let's say, reduced to the homozygous state? And then, because the risk of that, if that happens often, is that then good alleles that are in cyst to those bad alleles, you're never going to recover.
I'm not sure whether I understand the details. Maybe we can discuss this. But what we did, for example, we looked at, what I showed here are rather complex traits already. We looked at metabolites, used metabolites as traits, as our phenotypic traits, and there we found very interesting results. Really, we found the target genes underlying the metabolites directly. We could make a direct relationship, though that gives to me a lot of confidence that this method works very well and really produces a high map resolution. You really can get directly with this material to the candidate gene.
Okay, I'm Sujay from Indian Maize Program. It's always exciting to listen to you and go through your work. Once in your one clarification that you told that better to sample a few highly selected landraces with increased number. Now landrace in our gene bank is very huge. So how do we identify that this is the highly selected? I couldn't get it exactly. What do you mean by highly selected landrace?
Yeah, what I mean is, you are targeting the 80%. You are first targeting the 80% of the genetic variation which lies within a landrace, and for that reason, you don't need to worry too much about the 20%. Actually, if you would have double tablet line libraries available from, I would say, 5 landraces, you could cover, I would say, 98%. You would capture 98% of the genetic variation that is available in the thousands of landraces that are stored in the seed bank. Yeah? This is what I meant.
Sorry, my friend, I think we are running short of time. Please catch up during the tea time and then do that. So with this, please join me in thanking Dr. Melchinger.